screen.
Perfect. All right. I think Mrinal is going to lead us off here.
Yeah, sure. Yeah, so we're talking about Konzo. As we had a brief discussion earlier, it's related to cassava and the toxicity of the cyanogenic glycosides inside cassava. And Konzo is a sudden onset symmetric, non-progressive, reversible spastic paraparesis or quadriparesis due to selective upper motor neuron damage, and it's permanent. Once the kid, usually from Democratic Republic Congo, Nigeria, all these places get Konzo, and it's permanent. The damage is permanent, and usually they occur in outbreaks, and it's usually linked to droughts, and it's especially common in DRC in Congo. And usually it's related to a monotonous cassava diet. People usually consume mostly cassava, which is high in carb and less in nutrients, and the consumption of cassava is mostly done through improper processing techniques. And the reason is usually it's said to be due to these mainly due to cyanogenic glycosides like linamarin and otafzoline. And linamarin is most of the 95% of the cyanogenic glycosides are linamarin, and that's what causes the damage. Yeah. So what we did for the part of project is that we try to divide the whole picture into different aspects and try to see where we could make an intervention and make a change. So it could be at the level of the cassava plant, and it could be at the level of the processing of the cassava. It could be at the level of the environment, the level of the host or the treatment, and after that, any interventions like behavioral changes stuff. But in our case, what we focused mostly on was processing the cassava, because that's the place that can have the highest impact on how these people live.
Oh, thanks, Mrinal. So just to go over, as Mrinal mentioned, this is endemic, particularly in DR Congo, and there are different methods to process cassava. It's widely eaten around the world, but the primary method that's used in the affected areas is harvesting the tubers, soaking in water for 3 to 5 days, drying, pounding into flour, and then eating in a porridge, often called fufu. And so how we approach this, and I'll just want to say from the outset, really impressive to see all of the work that the other groups have done. What we were proposing here is a series of experiments. We haven't actually gotten to complete any of them, but nevertheless, we wanted to present this to you and get some feedback. So our hypothesis is that the pH of the cassava soaking water is impairing the potential detoxification that can be happening during that process, and therefore that this is negatively impacting cassava Konzo incidence and could potentially be improved with frugal materials to alter this pH. So a bit of the chemistry here: linamarin is the cyanogenic glycoside. Linamarin, while being soaked, is processed by an enzyme called linamarase. This produces acetocyanohydrin and ultimately cyanide, which is then eliminated during this soaking or processing process because it's a volatile compound. So in this case, linamarase is your friend because it's helping take this cyanogenic glycoside and produce cyanide that is then released. So the problem is that soaking ends up acidifying the water that the cassava is being soaked in, and low pH inhibits the action of this enzyme. And we think that water scarcity exacerbates this problem of acidification, because when there's less water available, there's more likelihood that that water will be reused over and over again, and these acidic compounds will build up and therefore prevent the effective detoxification. Sorry, I forget if this is my slide here. I think it still is. Yeah. So what we're proposing is a series of experiments to evaluate this process and see if we can produce a meaningful intervention, a meaningful low-cost intervention in this process. So the first experiment that we're planning is to emulate the cassava soaking and detoxification process. So obtaining tubers, dividing them into pieces, soaking 1/2 for 5 days, using low-cost test strips to monitor cyanide levels both in the soaking water, and then comparing cyanide that is retained in soaked cassava versus unsoaked cassava that is our input material, to evaluate the proportion of cyanide that is eliminated throughout the soaking process and use this as a foundation for further experiments. So I'll hand that off to the next speaker.
Yeah. So in the same line of processing cassava, so to the point that where it is more digestible, one of the way that we want to address in making it more effective in detoxifying is how can we actually adjust the pH that the cassava is soaking in. So as Dave has shown earlier in the cassava processing process, cassava is typically soaked from 3 to 5 days. And exactly this is what we're trying to emulate here. So we have chosen four different neutralizing agent and with the water as control. So we will add soak the water as well as various neutralized reagent at the same time with pieces of cassava. And with each of the days, we'll measure both the pH as well as the cyanic level. And so this is what we will expect to collect as data for figure 1 and B to see the cyanic level over time as well as the pH over time. But at the same time, what we care about is also the cost effectiveness of these neutralizing reagents, particularly at the site. And Dave, if you can move to the next slide. And then we realized actually the experimental setup that we're creating here is wonderful in addressing the pH level. However, the quantity of cassava and the toxicity level of the cassava might not, in the experimental setting in the beautiful lab of Palo Alto or Arizona, might not be comparable to that of Konzo. Congo. We actually have a scientist, Matt, who will be traveling to the DR Congo to the affected region. So what we plan to do some experiments there will be to test the pH level of those cassava soaking pits in both affected region and unaffected region, to get a sense of where the pH actually lies in the real sites. And we wanted to test one of the more effective reagent of neutralizing from experiment 2 and see how well we can deploy those reagents in neutralizing neutralized pH in the pit, and as well as like how costly it might be. So, and maybe at the same time, we want to research local availability of some more frugal and more reasonable neutralizing reagents in the affected region.
So the second hypothesis that we have is that cyanide content in cassava can be eliminated or decreased to tolerable levels by alternate methods of processing. So linamylase being an enzyme is inactivated by heat, and boiling is also expected to reduce the cyanogenic glucoside content in cassava. So we hope to include different steps in the processing and try to assay or measure the levels of cyanide in various methods of processing. So the experiment 4 that we are planning to do is to emulate different methods of cassava processing. We'll essentially peel the raw cassava tubers and process using each of the below mentioned methods. We'll dry and pound the cassava in one method. In the other one, we'll include a soaking step, and we'll measure the cyanide content at days 1, 3, and 5, and we'll follow it with drying and pounding. The third method that we are trying to do is to include a step of boiling for 20 minutes, which will be followed by drying and pounding. So the powder that is obtained after pounding through various methods of processing, we'll essentially mix them with room temperature water and make a paste out of it and test the amount of cyanide that is present using reagent strips. So we haven't essentially gotten the strips in our hand. That's why we haven't been able to conduct the methods per se. So we hope to get the reagent strips next week and hopefully share the results with the team by next week.
All right, so there are some long-term directions which you want to work upon. For example, we need to still work upon the whole detailed process of improving the detoxification soaking techniques. Also, we want to increase the accessibility of low-cost pH neutralizing agents, as well as we want to test how feasible is the incorporation of the process of boiling in the whole processing technique. And last but not the least, it is a soft measure of social mobilization for education on improved soaking technique, and as well as finding other alternative processes and methods. That's it. Thank you, Manu. Thank you, teaching team, and everybody who worked with us. Yes.
Quick round of questions. So just a quick question, Dave, Divyanshi, everybody. Do we know for sure that, the diffusion of the water getting in I know you guys had mentioned between 3 to 5 days, water is scarce, so sometimes people don't soak it long enough. Do you guys know absolutely that water does diffuse all the way to the core of the cassava? So Dave, when you've been in the field, have you ever taken one of the tubers and cut it in half and confirm what if a certain portion of the tuber is never receiving or
Right, right.
receiving? Yeah, what is the diffusion time scale for this object? Because we could very easily test that.
Yeah, absolutely. No, I think that's a good question, and I don't know the answer to that. Yeah, it's an enzymatic process that's happening, but a lot of the enzyme is in the peel, and it might be that that enzyme is only reaching the exterior. It might be that smaller pieces would be more effective. As we saw in some of the videos on this, making smaller pieces might not be super effective in the pits and being able to pull them out again, but that's a good question about whether it's even reaching the
I see.
center.
So you're saying the enzyme is in the peel, so it wouldn't make sense to peel it before soaking. But then on the other hand, there is a spatial distribution for that enzyme.
Yeah, I believe they do peel. I think the enzyme is both in the peel and the tuber, but I believe it's higher concentration in the peel.
I see. So by peeling effectively, you are suppressing that process too. But
Perhaps. I
it's a little bit
don't,
of a
yeah, I don't
challenge,
know. Yeah,
because
yeah.
if you
I know.
don't peel, you don't get effective diffusion in.
Right,
If
exactly.
you peel, you lose the enzyme.
Right, right.
Yeah, I don't know whether there is kind of an intermediate ground, which is like potatoes. Sometimes I like poking holes in a potato, like best of both worlds.
Yeah.
Yeah, Manu, yeah. Can I add something?
Yeah, go ahead, Mrinal.
Yeah, so the linamarin is actually inside some vacuoles that are the vacuoles inside the cell, the tuber cells,
Yeah.
and the linamarinase enzyme is in the cell wall of the meat cell wall. So when you crush it or when you grind it up, the cell wall, the condensates are broken down and they mix together. That's what happens when they grind it down. But when they try to put it in the fermenting pits, they actually put it in larger pieces and they don't ferment it often. Like, they don't ferment it enough for 3 to 5 days, and they have lack of water. So essentially, cutting the cassava into smaller pieces would definitely help. Yeah.
I see. Yeah. Any other questions? Henry was supposed to cook us some fufu, but he bailed on us, looks like, right? Or
Well, we tried. It didn't go so well.
oh, that's okay. We still wanted to see. Okay, he did attempt it. Oh, you attempted. Okay. Okay.
I think you have 5 pounds of flour, so you know you can always try again.
Okay. I think we're gonna